chapter 12 is pretty short. It goes pretty nicely with chapter 11. It's almost like an extension of chapter 11. In chapter 12, we're going to talk more about sex determination systems, human X and Y chromosomes, sex-linked genes, patterns of inheritance again, X inactivation, non-disjunction, and alteration of chromosome structure. So chromosomal sex determination systems are kind of interesting. The one I want you to know about for your exam is the one in mammals and some insects with the XX and XY system. So in that, females are XX or homogametic. They can only produce one kind of gametes. Their gametes are going to have an X chromosome, and males have an X and Y. So they're heterogametic. They can produce X sperm or Y sperm. And some mammals and insects have an XX or X0 system, or XO. So in this system, males lack the second X chromosome, so they only have one X chromosome. Then in reptiles, birds, and some insects, they have a ZW system. So the females here are heterogametic, which is different than we would be. And so they're either Z0 or ZW. And then males would be ZZ, so they would be homogametic. Then we have the haplodiploid system, where males are haploid and females are diploid. So, for example, in bees, the female has 32 chromosomes and the male has 16. So this female has 16 pairs of chromosomes to get to her 32 chromosomes. But again, I want you to focus on the XX and XY sex determination system, like in mammals, like ourselves. So as far as human X and Y chromosomes go, the X chromosome is much larger than the Y chromosome. The X chromosome carries much more information because it has more DNA sequences. It has over 1,400 genes. The Y chromosome only has around, 200 genes. An important region on the Y chromosome is the SRY gene, or the SRY gene locus. It's named for sex determining region of the Y chromosome, and it's named because in the absence of the SRY gene, the gonads develop into ovaries. However, sex determination is much more complicated than just this one gene. There's many biochemical, physiological, and anatomical features that distinguish males and females, and many genes are involved in this development, not just the SRY gene. And a gene that's located on a sex chromosome, whether it's on the X chromosome or the Y chromosome, is said to be a sex-linked gene. So if a trait is on the X chromosome of a human or mammal, we could say it's X-linked. If it's on the Y chromosome, then it would be Y-linked. And heterozygous females would be carriers, and males can be called homozygous for traits on the X chromosome because they only have one X chromosome. For X-linked Punnett squares, you need to use the sex chromosomes X and Y, and then use a superscript or an exponent to describe the trait. So, for example, X big N, X little n would be a heterozygous female, and X big N, Y would be a hemizygous male. And I feel like many students get into the habit of putting X and Y with every single Punnett square that they do after they learn about X and Y chromosomes, but don't do that. You only need to put X and Y chromosomes in the Punnett square if the trait is an X-linked or Y-linked trait. So be careful with that. And really, in our class, we're not going to deal with Y-linked traits. We're only going to talk about X-linked traits. So eye color in fruit flies is an example of an X-linked trait. The red eyes are dominant to white eyes. They use the W plus notation to denote red eyes. So in this example, we've got a red-eyed homozygous red-eyed female and a white-eyed male. The male can give an X chromosome with the white eye allele or a Y chromosome, and the female can only give a X chromosome with the red eye allele because that's what she's got 2 copies of. So in the F1 generation, we get all red-eyed females and all red-eyed males, but if we bred the F1 together to get the F2 generation, we get 2 red-eyed females and 1 red-eyed male and 1 white-eyed male. Here's just another example of an X-linked pedigree. In this case, this is for color blindness, which is a characteristic in humans that is carried on the X chromosome, and in this case, being unaffected or having normal color vision is dominant to being colorblind. So let's review with an X-linked example. So in humans, there's one gene for red-green colorblindness, like we said, and it's located on the X chromosome. Not having red-green colorblindness is dominant. Having red-green colorblindness is recessive. So could we have 2 parents that do not have red-green colorblindness have a red-green colorblind boy? So if we do that, we have to think about the possibilities of the mother. She could either be a carrier for red-green color blindness or be homozygous for not having red-green color blindness. So those are our two mom options there. She could either be X big B, X big B, or X big B, X little b. The dad, if he's not colorblind, he has to be X big B, Y. So that's our dad option. So if we fill out our Punnett squares here, there's our first one, and ta-da, there's our second one. And now we want to know, could we have a red-green colorblind boy? So to be a red-green colorblind boy, it would have to be X little b y, and we do have an X little b y in the second Punnett square. So that is a possibility if mom is a carrier for colorblindness. The next question asks if we could have two parents that do not have red-green colorblindness, and if they could have a red-green colorblind girl. To get a red-green colorblind girl, we'd have to have X little b, and that's simply not possible if her dad is not colorblind, because she gets one X chromosome from the mom and one X chromosome from the dad. So female mammals do an interesting thing. They can do something called X inactivation. So in all of their cells, they have two X chromosomes, and in each cell, so far we think it's randomly, an X chromosome is inactivated during embryonic development. This inactive X condenses into something called a Barr body, and so the female, if she's heterozygous for a particular gene located on the X chromosome, you're a mosaic for that character because it depends which chromosome is turned on or off in that particular cell. So what is going on here? We've got cats that could either be black, orange, or female cats that could be both black and orange. So if we look, you can see that the X chromosome is what's carrying this either black or orange coloration, and the females, since we could have a female cat that could be both black and orange, that is an example of X inactivation. So if the female is heterozygous for those colors on her X chromosome, the different cells in her body will turn them on or off, and whichever cells turned off a particular chromosome will have a particular color there. So there's several different ways chromosomes can have their structure altered. A breakage of a chromosome can lead to 4 different types of changes in chromosome structure, and these are, I think really they're pretty well named for what they are. A deletion removes a segment from a chromosome, a duplication just repeats a segment, an inversion flips the orientation of a segment within a chromosome, and a translocation moves a segment from one chromosome to another. So we can also have abnormalities with chromosome number. In non-disjunction, pairs of homologous chromosomes don't separate normally during meiosis. So as a result, one gamete receives 2 of the same type of chromosome, and another gamete is going to receive no copy of that particular chromosome. Aneuploidy results from fertilization involving gametes in which non-disjunction occurred. So offspring with this condition have an abnormal number of a particular chromosome. For example, a monosomic zygote has only 1 copy of a particular chromosome, and a trisomic zygote has 3 copies of a particular chromosome. Polyploidy, on the other hand, happens when an organism has 2 or more complete sets of chromosomes, right? If we look at the words like mono, 1, tri, 3, poly, many. So triploidy is when you have 3 entire sets of chromosomes, or tetraploidy is when you have 4 entire sets of chromosomes. So triploidy and tetraploidy aren't really things that are going to happen in humans, but they can occur in some organisms. So here's just an example of showing you where non-disjunction can occur and how it's a little bit different if it happens in meiosis 1 or meiosis 2. So either way, we can end up with cells that have an abnormal or atypical chromosome number. So let's match the example to the term. So Klinefelter syndrome is when there's an extra chromosome and it produces XXY individuals. So that's going to be a trisomy because there's 3 sex chromosomes there. Number 16, modern strawberries have 8 complete sets of chromosomes. So that's going to be polyploidy, right? 8 is more than 2, so 8 is many, so polyploidy. Turner syndrome produces females with 1 X chromosome. So if they have 1 X chromosome, 1 mono, so that's monosomy. And then Down syndrome is a condition that results from 3 copies of chromosome 21. So 3, that would be trisomy. So chapter 12 is really kind of like almost an extension of chapter 11. We looked at patterns of inheritance with sex chromosomes. In particular, I said pay attention to human X and Y chromosomes and really pay attention to X-linked traits. We also talked about X inactivation, where female mammals turn off one of the X chromosomes in their cells, and so they have a mosaic of characteristics depending on which X chromosome they inactivated in their cells. We also talked about non-disjunction, where the chromosomes don't divide appropriately in meiosis, and alterations in chromosome structure. So that's it for chapter 12.